Hi, I'm Ushma Neal, one of the hosts of The Wild Types. This is a new video interview series that I'm producing together with the Film Annex. My friend Rich and I thought about why people don't think science is interesting. Why isn't it more in the news? Why are our celebrities not more scientists? Through this video interview series, we hope to introduce you to some of the most compelling and fascinating people that we know who also happen to be scientists. I hope you'll tune in to watch some of these episodes, maybe learn something, and make a cool friend. Hi, I'm Ushma Neal, and I'm here to introduce you to my colleague and friend Richard White of Memorial Sloan Kettering Cancer Center. Now, what do we call someone who hangs out in a lab all day? And it's not a trick question, a lab rat. Well, today I'm going to introduce you to a lab fish. Rich studies cancer and metastasis in zebrafish, and I think you'll find his interesting path through science entertaining. So Rich, can you tell me a little bit about what you do?
I'm I guess a cancer biologist, so I'm a MD, PhD, and so that means I have a medical degree and a research degree, but I mainly do research, and I still see patients sometimes, but I'm primarily a researcher. And I do a kind of strange type of research for a cancer biologist, which is that I study cancer in a fish called the zebrafish. I know this sounds kind of weird, but it's sort of like if you want to study cancer on a very large scale and really understand all the different ways that people can get cancer, well, one way of going at that question is you have to study lots and lots of variables, and studying cancer in humans and in mice is pretty tough, but fish are small, and it turns out that fish get cancer spontaneously in the wild, and they're really easy to reproduce. And so if we want to study cancer in its largest sense of the word, we can study hundreds of thousands or millions of fish in a way that you can never study it, in any other system. And so the way I think about the fish is it's like having your mother, father, brother, sister, uncle, aunt, and every other member of your family in one room that's basically the size of a pretty fair size aquarium. And you could never do that in any other thing. So it'd be like going back to the 1500s and capturing your whole family's history of cancer, but in one room. And so that's kind of why we study fish. The other reason I guess we study fish is, in addition to being small and cheap and all those other things, is that we made a type of fish that's transparent. And so this lets us study cancer not when it's too late and it's already spread, but figuring out how does cancer start? Because that's the thing you can never see in a human. You can't see cancer before it's obvious. And in fact, by the time most people present with cancer, they already have billions of cancer cells. We can see cancer when it's starting at a single cell and figure out what happens along that entire path of the cancer. And so by having a really transparent fish that lets us see it, coupled with this huge numbers of fish, it's just this really unique way of studying cancer that you can't do in any other way.
All right, so Rich, this is where zebrafish come to have a good time?
Where they come to, get it on. And so when I was in Boston, we kind of had this idea, another student, that fish like to mate in shallow areas. That's kind of where they naturally mate. And so they had the idea, a bunch of people, what if we put a bunch of fish in here and then simply lifted them up so that they were forced to mate on this little shallow platform like that. And that's exactly what happens. And then you kind of force them to be in a shallow area, and then the eggs fall through here, and then you collect them on the bottom. And even though we thought this was totally nuts, it completely works. And so that's how you can get 20,000 fish a day.
The primary cancer you're studying is melanoma. Why?
Most good science stories, it's like a combination of luck and timing and then interest. When we started to think about how can we study cancer in a fish, which seemed kind of like a ridiculous question when we started it, we kind of just said, well, what cancers do fish normally get? And one of the cancers that fish normally get actually is melanoma. And in fact, the reason zebrafish are called zebrafish is because they have stripes like a zebra. Those black stripes turn out to be identical, nearly identical to actually the cells that give us, humans, our pigmentation. So people who are black and white and all colors in between have varying amounts of those cells called melanocytes, which are pigment cells. And fish turn out to have melanocytes the same way humans do. And it was really obvious to us, well, if they have those cells already and they already get a type of cancer associated with those pigment cells called melanoma, it just seemed like the most logical place to start because they had all the right machinery to go into it, the right cells, and they already spontaneously developed that cancer called melanoma. So melanoma is quite simply just a cancer of melanocytes, which are just pigment cells, and that's what it is.
So we're down looking at the actual fish, and Rich, can you tell us what the difference is between these two tanks?
Sure. So this is a tank of pretty normal zebrafish. They're a little bit young, but they're getting towards adulthood. And as you can see, these are just swimming. We usually keep 10 or somewhere between 10 and 100 fish per tank. They have little stripes on them, and those are just normal fish. And these are fish around the same exact age that are the transparent fish, which we call Casper, that are essentially exactly the same. We call them Casper because they're kind of like ghost-like in a way, although I have to say I didn't name them. Somebody else in my lab named them. And so they function just about the same, except here there's no stripes to begin with. So we sort of add the color back to the fish. And in this case, like these fish, you can't see this with the naked eye, but these fish already have tumors inside that are growing, but they're fluorescently labeled. So that way they're invisible to the naked eye, but we know that they're already inside.
Well, thank you so much, Rich, for taking the time to chat with me today, and I hope that everyone will tune in for the next one.